Hello, my name is Dr. Charles Knight, and I teach a course called Bio 414 Evolution. It's our upper division evolution course here at Cal Poly in San Luis Obispo, California. And I'm recording a series of lectures that I'm giving to that class that I'm calling 10 Lectures on Evolution. And I'm posting them on YouTube so that you can view them. These are all presentations about recent scientific literature, papers that I've selected for this course that I think illustrate important concepts in evolutionary biology. So check back often. I'll be posting weekly or so new lectures about evolutionary biology on this channel, and enjoy this one. This paper is called Human-Driven Evolution of Color in a Stonefly Mimic. It was published on October 25th of 2024 in Science magazine. The details are up there for the publication. Before we get started on this paper, I wanted to highlight one aspect of one figure that I think is really important to understand this whole paper. And I'm just going to do that all the way up front here before I get into my take-home points and reading the abstract and all the normal things I do. But this is a classic story of Batesian mimicry, where there's a model, and the model is poisonous. Birds and bats and fish probably also don't like to eat this model. It's poisonous. In New Zealand, where this paper takes place, there's a mimic of that model, and there's a non-mimic too. The important component, I think the reason I'm pointing this out early is that you got to pay attention to these tail hairs. I'm calling them tail hairs. They're not called tail hairs. They're called cerci. If you're an entomologist, you see the cerci in the model are very short. They're not long like in the mimic. In the non-model mimic, there's also long cerci. So these images are used over and over again in the figures, and when you see the insect without long cerci, that's the model that's poisonous. Also, red text is associated with the model, which is poisonous, and I also put these emojis up there. Yucky, making your stomach hurt, versus yummy. The non-mimic and the mimic are both very palatable and edible for birds and bats. Now I'll go into what I call my scientist vignette. Stephen Nee was the lead author on this paper, along with Jonathan Waters and Graham McCullough. Jonathan, I think, was the PI. A lot of the backstory we don't get through the literature. I don't know these people. But what I could deduce is that there's a lot of undergraduates that are associated with this study. So these are pictures of Graham and Jonathan. I chose not to post a picture of Stephen. I think he's an undergraduate, and he might have been surprised that this would have been picked up like this, so I only posted pictures here of Jonathan and Graham. They are readily searchable on the internet, and both have now labs in New Zealand that are accepting students. They'd love to hear from you, I assume, maybe. But the paper takes place in New Zealand, a beautiful place down on the South Island in this province called Otago, and Dunedin is where the university is, I believe, and it looks beautiful down there. I'd love to go visit sometime. So the take-home points that I have for you for this paper is that Batesian mimicry is happening in stoneflies in New Zealand. I already said that. That's an important take-home point, and I'll talk a little bit more about Batesian mimicry in a second. Humans have deforested much of New Zealand. Museum and environmental DNA analyses have shown that there's a change in frequency of the model species, and as soon as we start to say change in frequency, it should raise your attention, because you know that our definition of evolution is a change in allele frequency. So this is another example of evolution in action, and fairly recent in New Zealand. Mimic stoneflies were protected from avian predators. That is, they were mimicking the model that was poisonous, but had lower reproductive output. It actually had a cost, and so there's a trade-off of producing this dark coloration in the mimic species. This is a little bit complicated, and I'll get into that a little bit later. Logging has certainly decreased the frequency of the model and therefore the mimic too, because it changes the habitat considerably, and the model prefers dark, cool, clean forest streams, and the hydrologic environment has changed quite a bit since logging has predominated. A last take-home point is that examining natural variation in non-model species like these stoneflies can be used to help identify genes or alleles underlying phenotypic traits. In this case, they identified a gene called ebony through natural variation in the stonefly population. They did a whole lot of genetics to get there, and it's pretty cool that in a pretty traditional evolutionary story involving testing fitness effects, they're also able to get down to the level of the gene, including lots of undergraduates. So kudos to Jonathan Waters and Graham McCullough for pulling this off with a bunch of other students, and I hope I got that story right. But it's a great paper. So now, as I do, I'm going to read the abstract slowly, just so that we all are kind of starting at the place where the authors intend us to. That is, having read the abstract. So here we go. Rapid adaptation is thought to be critical for the survival of species under global change, but our understanding of human-induced evolution in the wild remains limited. We show that widespread deforestation has underpinned repeated color shifts in wild insect populations. Specifically, loss of forest has led to color changes across lineages that mimic the warning coloration of a toxic forest stonefly. Predation experiments suggest that the relative fitness of color phenotypes varies between forested and deforested habitats. Genomic and coloration analyses of 1,200 specimens show repeated selection at the ebony locus controlling color polymorphism across lineages. These findings represent an example of human-driven evolution linked to altered species interactions, highlighting the possibility for populations to adapt rapidly in the wake of sudden environmental change. I think that was a pretty well written, very well written abstract that didn't have a lot of jargon in it. There's a few that I'm going to highlight here, partly for my students because we're not using a textbook in this class, so I like to highlight certain phrases and words that would be a part of a normal textbook kind of presentation of evolution, and anything that's a little bit specific. So I'm going to talk about the ebony locus, fitness, and phenotypes as take-home words, and just also underline this phrase that evolution is happening rapidly in the wake of sudden environmental change in New Zealand, and that's happening worldwide. And evolution is happening and may, it's going to continue to occur with climate change, and always has too. So I do this glossary notes to kind of introduce these terms. First, I should say fitness. Fitness, fitness, fitness. The relative number of offspring of an individual compared to the most fecund or individual in the population. You can even compare populations. Fitness is one of those words that's used a lot in evolutionary biology, so I think it's important to consider the definition of it. It's all relative. The individual that produces the most number of offspring is the most fit, no matter how many they produce. Every other individual in the population is ranked in relation to how many offspring they produced versus the most fit one. So it's a relative measure of offspring in a population. The phenotype is the outward appearance or physiology of an organism, and we'll use that word over and over again in this course. The locus is the location or locations of the genetic basis or the genes that are controlling a trait, and the locations refer to actual physical locations on a chromosome or in the genome. And ebony is a candidate gene that they tested for relation to black coloration. This candidate gene had been discovered previously in Drosophila and other organisms, and shown to be associated with dark coloration and dark spots in some cases, and it just, it was a shot in the dark, and it came out to be a significant hit in their genomic analyses. So I always do what I call textbook tie-ins. I guess partly, again, I'm not using a textbook in this course, but instead kind of just bringing out a few important parts of the textbook that I used to use. And I thought that I'd talk about Darwinian fitness here. Darwin thought of individuals whose variant traits improve their chances of living and procreating as having been naturally selected, just as a tomato with its large fruits is artificially selected as a seed stock by a farmer. It is crucial to recognize, however, that during natural selection there is no conscious intent and no ultimate goal in mind. During natural selection, it just happens to be the case that individuals with a particular variant trait do better. Darwin described individuals who are better at surviving and reproducing and whose offspring make up a greater percentage of the population in the next generation as being more fit. Darwin described it this way. He thus gave the everyday English words fit and fitness new meanings. Darwin did that, and we call it Darwinian fitness, and it's an individual's ability to survive and reproduce, as I just explained. The individuals that are the fittest in the Darwinian sense are not always the ones we ordinarily think of being the most physically fit. But I'm going to leave it there. Fitness is kind of intuitive, but I want to make sure it's intuitive to you going forward. So fitness is mentioned on 150 pages in the text that we kind of are using as a reference text. So it's a very frequent word. The backstory here about this study is the environmental change that's happened in New Zealand, and it's not unique to New Zealand. Most places that have been colonized by Homo sapiens at some point experience quite a bit of deforestation. New Zealand hasn't escaped that. There's been significant deforestation since 1840s and even before that. So this is a map showing this kind of deforestation. If we look just on the North Island, it's extensive, 1860 to 1939. It's like, wow, they really deforested a lot of the island. They was the colonizers that came after James Cook first kind of contacted in 1769, and in the 1830s, British and other nationalities really started to settle there. But the Mary actually started this deforestation 1,000 years ago or so, because they were recent colonists too at one point. The deforestation leads to land use transformation and pasture formation, and then obviously significant erosion. And this has changed the hydrologic environment for stoneflies in the native forests and landscapes of New Zealand. Stoneflies look like this. The larvae are on the upper left and their adult form is on the lower right. They're very favored by fish and also avian predators, and they're often mimicked by anglers to try to catch fish. Stoneflies are common worldwide, and this is a figure from one of the references from the paper showing the phylogeny of some of the stoneflies and their geographic locations. The colors correspond to the geographic locations. You can see the northern hemisphere in red, and the southern hemisphere are the other colors. Australia is yellow, New Zealand's black, and so in some cases you might expect the yellow and the black to go together because they're fairly close together. South America's blue, that's much further away, and South Africa is also in green. So by looking at this figure carefully, you can identify instances where dispersal was probably important, like getting all the way over to South Africa was probably a dispersal event, and also getting to the northern hemisphere is also a long-term dispersal event, probably a long-distance dispersal. But the divergence between Australian and New Zealand species could have easily been due to vicariance, just the separation of those land masses. You can see South America is snuggled up in there quite a bit too. Leads to the question, is this vicariance or are those dispersal events? These are interesting phylogeographic questions that this paper doesn't go into. So I'm going to leave that there. Just a little bit of natural history. This is a different species of stonefly with its own cerci. It's not like the model species. So this is another figure from our textbook that this shows dispersal quite well, pretty intuitive. There's a vacant island. Individuals move over there. That's a dispersal event. And then vicariance is when this island, perhaps just by geologic forces, is separated, and the existing populations then can evolve just by random chance or by natural selection that's different on the two islands. And after a period of time, they would accumulate enough genetic differences to be identified as different lineages or different clades, and that may be playing a role too. So I just want to make sure that everybody's on board with this word vicariance, which is the splitting of a population's former range into two or more isolated patches. Again, that might lead to speciation, and Ernst Mayr, one of the leading figures in the modern synthesis of evolutionary biology, really thought that vicariance explained a lot of speciation. Well, I mean, it is a very important part, for sure. So the river environment has changed dramatically since deforestation, and this is actually one of the first figures in the paper. I think it's 1C, where we see the forested creeks on the left and the deforested on the right. At first pass, it may not seem that different, but the thermal environment is quite different, and even the suspended sediment in the river is quite different. And these environments are actually very different, and the avian predators are actually much different too. The birds much prefer the forested habitat. Maybe surprisingly, I'm used to having seeing lots of raptors in open areas, but bird predation is much higher in the forested streams versus the deforested streams. So the habitat has changed, and the cause of that is human deforestation. So that's an important starting point. I'll take you back to our mimics and non-mimics and model. The model, remember, does not have those long cerci, and it's unpalatable. And this is an example of Batesian mimicry. Remember, Henry Walter Bates wrote a letter after the publication of The Origin of Species, which he read down in South America, and he thought to himself, I have some data that might shed light on Darwin's theory. And he wrote a letter to Darwin that said that after he read The Origin of Species, and it involves this mimicry of a non-palatable, or mimicry of a palatable species to a non-palatable or a poisonous species. The same situation we're talking about here. Bateson had observed back in the 1860s and 70s in South America, and he thought that this might help Darwin in his gathering of support for evolution by natural selection. So in this study, they measured melanism in these stoneflies, and they did this with a spectrophotometer, and therefore they had rapid phenotyping. They could send lots of stoneflies underneath the spectrophotometer and measure their melanism index. I'm probably making it sound easier than it was, but we can see that the model and the mimic and the non-mimic kind of parse out in different areas of this melanism spectra. And it'll be shown better even on the next figure, where the different genotypes have different spectra. But this is figure 1b, and it begins this just measuring the phenotype of these stoneflies, and so that they can measure thousands of them. A little bit differently presented, same information, same melanism index. We now see the different genotypes, the different species. Remember, the model that's up in red is a different genus than the mimics, which are a different genotype and different species actually. And within that species, we have three different genotypes. We have a recessive homozygote a, which is the mimic, which has the black coloration, and then we have heterozygotes, and then we have homozygote dominant, which are the non-mimics, which are brownish. Both the mimic and the non-mimic are palatable, but they also separate out on this melanism index, so it makes it very useful to be able to rapidly genotype these individual bugs. The distribution of the bugs has changed with deforestation. Right now, environmental DNA analyses, really, they used environmental DNA analyses to figure out where these bugs live, especially the mottle, which is not as common because of deforestation. The mottle tends to be in forested habitats, which are becoming less and less common. Instead, they did stream sampling for environmental DNA just to probe whether or not this mottle species was in those streams, and they only found it in these streams, which are in black. The whites are all places where they sampled but didn't find the mottle species. Previously, as we'll see next, they were there, but right now from their environmental DNA, they only see it in the darker green patches, which are the forested habitat. Of course, there's some places where they didn't pick it up, but the correlation is pretty strong here. This is in a supplementary figure for the paper, and they guess they just ran out of space. They couldn't include it. The next figure shows museum museum science, where they looked for records from notebooks and also collections for the location of where these insects were found. And what they found historically is that the mottle was more widespread in New Zealand, and also still associated with the forested habitat. However, I think this green coloration is current day forestation, but suffice to say, they did some museum science and some environmental DNA to study the distribution of this stonefly in New Zealand as well, to begin to understand what's going on. So figure 1 is starting to explain where these mottles and mimics and non-mimics live. The mottle is the poisonous one, the mimic and the non-mimic are palatable, and the mottle prefers the forest habitat. And in the forest habitat, the mimic is not predated on, the mimic and the mottle. Notice the red circle is the poisonous mottle. Birds, red X, are not predating on those mottles. They're also not predating on the mimics, which are below. They're not in a box. They're in you see the longer cerci, but they're still black. Those are the mottles. Those are the mimics of the mottles. They're not getting eaten either because they're black. But the non-mimics, the brown ones, are getting eaten. And I'll show you how they could tell that. They had a really cool experiment they did to show that. In the deforested habitat, the mottles are rare, and therefore the mimics are too. But the non-mimics are there. But in general, predation is pretty rare in these deforested habitat because the predators just aren't there, and the mottles aren't there and the mimics aren't there. Generally not as good of a habitat for stoneflies, but they can still test for this, and we'll see this cool experiment that they did. In general, what we see is the birds are rare, there's not predation on the mottles, there's not predation on the non-mimics, and there's not much mimicry because the mottles aren't there. So the system is kind of broken in a way. But we wonder, why are the mottles and mimic rare in these habitats? I think partly, again, is due to the mottle preferring these cool, dark streams. They feed on coarse, woody debris in the stream. The hydrology of deforested habitat streams is quite different, and there's less coarse, woody debris, more sediment, and they're just not it's different. They don't enjoy that habitat. So this is the overall figure 1C, and so I just kind of parsed it out for you there. But what we can tell is that the mimic frequency is much higher in forested habitat than in deforested habitat. And we look at the melanism index here, and these are just different collections, but the point here is that the the mimic frequency is much higher in the forested habitat because the model is there. In the deforested habitat, the mimics are much less frequent. So deforestation changed allele frequency. And I just want to point that out because remember, our best definition for evolution is a change in allele frequency. We know that the mimicry is caused by a genetic change. We know from their melanism measurements that the genotype is correlated with the melanism index and therefore mimicry. And there's been a change in allele frequency, and they showed it simply by just measuring this, but they have done more detailed genetics too, and other experiments also, which are really clever. The current populations on the South Island down there by Dunedin are separated into 4 populations, or 4 clades, clade 1, clade 2, clade 3, and clade 4. And we'll see this on the next slide where they genetically co-segregate amongst those 4 clades. And what's interesting is that the mimics and non-mimics segregate together within each of these clades. Within each of the clades, there's forested and deforested habitat, but it's not the case that the bugs in the forested habitat are all more closely related, or all within a subclade area more close. Well, I should say the bugs in a subclade are all more closely related, whether they're in forested or deforested habitat. And we'll see that on the next slide. This is a PCA analysis, principal component analysis, of lots of different information, I assume, including genetic SNPs. 78,360 SNPs are grouping individuals from 4 subpopulations together. So they did a PCA of the SNPs, which is interesting. If the investigators were here, I'd want to say, well, why didn't you just use a phylogeny to do this? And I'm sure they have a good answer for why they didn't do that. It reminds me of the old way of doing taxonomy by PCA instead of using, homologous looking for similarities by common descent instead of just using all the traits together. I'm curious why they did a PCA, but it turns out to be a very convincing story that these 4 subclades all cluster together. The dotted line surrounds them, kind of a statistical clustering, and we see the mimic and non-mimic all grouping together within those subclades. So they're migrating to these different areas by dispersal, because there hasn't been really chance, maybe vicarious, but I'm thinking more likely dispersal. But then they evolve mimicry repeatedly in these different areas, which is pretty phenomenal. There's been convergent evolution of Batesian mimicry in these different subclade groups. Convergent evolution of Batesian mimicry in these 4 groups independently. The big question about convergent evolution is there a common genetic basis for these instances of convergent evolution? And we're getting closer in this paper to seeing that, well, maybe ebony, the locus, has been selected repeatedly for increased expression or changes in expression that lead to black coloration. So this scientist did a really cool experiment where they made models of the mimic and the non-mimic and the model. So they made the mimic and the non-mimic models. It's hard, the words are hard. These are fake. They made this fake presentation in clay of the mimic. Look at the cerci. The cerci are long on the black one. That's the mimic. And the non-mimic, which is brown, which is fake. And then they placed these in the creek environment and looked at predation on these fake bugs. I'm being careful not to use the word model, because remember, the model is the different bug that doesn't have the long cerci. These are fake clay, look, I wanted to say model, but I don't want to confuse you. These are clay representations of the mimic and the non-mimic. So they looked at predation, predator attacks in the forested habitat. The mimic had much lower predation attacks compared to the non-mimic. So the birds know to stay away from the mimic. And so non-mimics are preferred prey in the forested habitat. In the deforested habitat, there's not a significant difference, but predation is overall lower there, and remember the model is also less frequent there as well. So that's a pretty cool experiment, fun. I wonder who made those models. I think maybe collaboration with the art department, but that's really neat that they were able to pull that off. Remember that the mimic frequency is low in the deforested habitat, but it's higher in the forested habitat. And the question is, if bird predation is low in deforested sites, why are the mimics also less common? Well, they also did something really cool, because remember in Darwin's 4 postulates of evolution by natural selection, there must be a difference in reproduction for there to really be a fitness difference. And so what they did is they looked at ovarian mass of the mimic compared to the non-mimic. And the details here I didn't fully get into. I'm not sure, I can't remember if this was in a controlled environment or if it was in the stream environment, but the mimics have a much lower ovarian mass, implying that they have a lower reproductive output and perhaps lower fitness than the non-mimics. And it was a significant difference. So we're seeing differences in reproductive performance of the mimics and non-mimics, indicating that there's an evolutionary trade-off. The mimics have less ovarian mass, presumably fewer offspring, and therefore lower fitness compared to the non-mimics, and this points to an evolutionary trade-off, and we're just hitting all the high notes of evolutionary biology right here in this one paper. These days, the question of how the mimetic form evolved is of much interest, and when we think of how, we're thinking about the genetics of how it's produced, and so the scientists used that big SNP data set that they used, 78,000 SNPs, and asked the question, which SNPs are more associated with the black coloration? That's called, in general, a genome-wide association study. In a way, I don't know if they called it that here, but then they added in this information about one of their SNPs, or maybe a few of them, that are close to the ebony locus, and they wonder, is that ebony locus associated with the mimetic form? And as it turns out, the answer is yes. The ebony locus is much higher on this scale on the left. I'm sorry I didn't put the axis label in there, but I think it's something like the linkage disequilibrium frequency, which is the logarithm of the odds, and I've just lost you completely. It's just higher. It's higher, meaning it's much more associated with the dark coloration. And we see the genomic position of all 78,000 SNPs here. Maybe this is just one of the chromosomes, or maybe it's the whole genome, I don't know. But ebony is off the charts in terms of perhaps being the genetic basis that's co-segregating with mimicry. And that's a neat addition for this paper. So I'm going to do my take-home points once again, and then we'll be done. Batesian mimicry is happening in stoneflies in New Zealand. Humans have deforested much of New Zealand. Museum and eDNA analyses show a change in the frequency of the model species in this Batesian mimicry system that they're studying, as well as the mimics. Mimic stoneflies were protected from avian predators when the model was there, but had lower reproductive output, and therefore there is an evolutionary trade-off. And logging decreased the frequency of the model and the mimics, and therefore led to a change in allele frequency in this population. And then, by studying or examining natural variation in non-model species in nature, that can be used to help identify genes or alleles that are underlying phenotypic traits. So we're in this new era of evolutionary biology where we can do our classic evolutionary studies, which I think this paper did a fantastic job of doing, and then pull in the modern genetic basis for these phenotypic traits too. Kudos to this whole team that produced a phenomenal paper in Science just recently. That's the end.